Good afternoon and welcome to the live Mastermind tutorial webinar. I'm Candace Chapman, Director of Marketing for Genomenon, and I'll be your host today. Dr. Mark Kiel is with us and he will show you how you can use the Mastermind Genomic Search Engine to quickly find key articles on disease-gene relationships and variant-specific searches. He will also show you how to narrow your search to specific fields of interest with different use cases that may apply to you. As you're watching, please feel free to submit questions to Dr. Kiel through the chat window on the right of your GoToWebinar control panel or at Twitter at Genomenon. And we'll have questions after the walkthrough. So without further ado, I'm going to introduce our speaker. Dr. Kiel is a founder and chief science officer here at Genomenon, where he oversees the company's scientific direction and product development. Prior to starting Genomenon, Mark completed his residency in clinical pathology in 2014 at the University of Michigan. While at Michigan, he completed a fellowship in molecular diagnostics and devised the informatics framework for clinical next-generation sequencing in the molecular diagnostics laboratory. During his doctoral studies, he made groundbreaking contributions to the study of hematopoietic stem cells, for which he was awarded the Weintraub International Graduate Student Award and the ProQuest Distinguished Dissertation Award. And while a postdoctoral researcher, he made significant contributions to the field of hematopathology, including genomic profiling of lymphoid malignancies, for which he was awarded the Benjamin Castleman Award. So without further ado, Mark, take it away.
Thanks, Candace, and welcome everyone. Today, for the tutorial, I'm going to give a very brief, broad overview of Mastermind, its contents, and its potential uses. I'm going to walk through some of the components of setting up the Mastermind software for new users. And then I'm going to go through a series of use cases which are intended to highlight some of the features and the technology and the data aspects of the Mastermind database. So to get started, let's talk about what Mastermind is. The goal of Mastermind is to organize genetic and genomic information from the medical literature. To do this, Genomenon and our team seek out from the 30 million titles and abstracts in PubMed and all of the new submissions on a nightly basis, looking for genetic or genomic content by indexing those titles and abstracts and metadata to identify those PubMed IDs or papers that contain genes, diseases, or genetic and genomic keywords. When we've identified that one of these papers contains relevant content for our users, we seek to index the full text and in many cases the supplemental information and insert that new information into the Mastermind database on a weekly basis. To date, we have over 6 million full text articles and more than 50,000 supplemental materials from highly prioritized references. And again, those numbers continue to grow on a weekly basis. And what Mastermind's infrastructure indexes from those papers and files is the mention of any one of thousands of different diseases, building on the medical subject heading or MESH disease ontology, or any one of the tens of thousands of genes in the human genome based on the HGNC conventional symbology for those genes, as well as a great variety of different synonyms or acronyms or deprecated names for any of those genes. And once we've identified disease or gene in a reference, we seek out to index the full text and or supplemental data. And we look through that material with our technical process for the mention of any one of billions of possible genetic variants, no matter how they may be described by the authors of those works at the cDNA level or the protein level, using HGVS nomenclature or not, or otherwise described using RSIDs. So Mastermind, the interface, which we'll spend the majority of this tutorial walking through, was originally intended for clinical purposes. As Candace alluded to, that is my background. That was the the original inspiration for developing the Mastermind search engine, given the needs that I faced as a molecular genetic pathologist to identify this reference material. But increasingly, we've found with our broadening user base, it has wider applications and is generally applicable to anyone looking at next generation sequencing data or otherwise genetic information in general. So the original purpose for Mastermind in clinical circumstances was required that Genomenon focus initially on maximizing the sensitivity of our database so that a user going to the interface would be assured of the maximally sensitive search results to ensure that no content was missed in the pursuit of diagnosing and treating their patients. And increasingly, as we've gotten a very high level of sensitivity for our searches, the highest that we've seen compared to any of our commercial competitors, we're able now to hone in more on the specificity of those search results in ways that I'll showcase here in a moment. So that's the overview of Mastermind. What I want to next do is to spend a couple of minutes talking about the setup of Mastermind. I'm landing here on the Genomenon homepage at www.genomenon.com. This is the landing page. For those of you who have not yet signed up, I'll direct your attention to the bottom of this page, and it appears throughout the rest of the website, where you can try the Mastermind software for free by clicking on this red button. I'm already obviously a account holder, and so I'll show you what that window looks like when you perform that search. This is a Chrome incognito window. So if I click on that red button as a naive user, I'm prompted to sign up for Mastermind Access with some required fields. There's an end user license agreement to agree to and confirmation that you are a healthcare professional or researcher. And then you click the sign up button, and that will trigger our engagement with you to give you your account, give you the necessary information via email to get your account set up, as well as to change or reconfigure your password. So one of the first things to highlight when you get your emails after initially signing up as a new user is the only required aspect of setup is to install the Google Chrome extension that goes along with the Mastermind software. So you can access this extension through the link that's provided in the email that you initially receive after signing up, or otherwise go to the Chrome Web Store or Chrome extension. If you want to look it up in a Google search, you'll be taken to this page. If you perform a search on Genomenon, as I have done here, or otherwise Mastermind extension, here is the Chrome extension. Chrome is the preferred browser for interacting with the Mastermind software. The purpose of the extension is solely to allow users who have access to the full text PDF content to see that content in the Mastermind app itself. And so it's very lightweight, it's easy to install, it takes about 5 seconds, and then once you've got your login credentials, you're ready to begin using the Mastermind software. Before I jump into that aspect of the webinar here, the tutorial aspect, I want to talk through a couple of the different ways that we give out this information to our free users as well as to our professional edition users. The free edition, which I'm highlighting here under the Mastermind product and services dropdown, is available indefinitely to any users who wish to sign up. Any such users are afforded the ability to access the professional edition, which is available to them for 30 days after their initial sign up. But then after that 30 days, their account will convert to free, and they'll be able to use Mastermind for many years hence. The professional edition differs from the free edition in a couple of ways, but not in terms of the data content. So the data content and the frequency of update is the same between the free and professional edition. The major difference is that the free edition has a cap on the number of searches that can be performed per week. And the professional edition is uncapped. And there's a couple of other salient differences between the two that you'll get accustomed to as you go through your free versus professional edition use of Mastermind. So once you get the welcome email, which is the primary way that we communicate with our user base to provide information about software updates, as well as to provide a monthly newsletter, you can proceed to the Mastermind homepage itself, which I'm showing here. Just to orient you a bit, there's the toolbar at the top where you can always return to home here to return to the Mastermind homepage. We encourage people to contact us with any questions they have. If they found anything particularly valuable about one of the searches that they've performed, that's very meaningful to us. If they have what they think is a novel use case that they'd like to discuss with us, we'd be very interested to hear from them, as well as having any questions about some of the content, either from comparison work that they've done or from real-time clinical searching that they've performed. Any kinds of questions like that will eventually reach my desk, and I look through those and greatly appreciate any interaction we have with our users. A couple of features to talk about, in addition to the way that we give this information to our users, Mastermind Alerts is a way for you to be apprised of newly published literature before or in the absence of any need to search the Mastermind database. If you have a variant or 1,000 variants that you're interested in keeping track of, whether they've been published in the medical literature, Mastermind Alerts is a way for you to receive that information by email and then be taken to the Mastermind software through links that are provided in those periodically updated emails, whether weekly, monthly, or quarterly. I won't spend too much time talking about the batch processing capabilities that Mastermind has, but for those users who have more high throughput needs, we have batch VCF processing capability, as well as various APIs that can provide more of this information that I'll show you in the Mastermind user interface in a much higher throughput for labs with their own bioinformatic team or individuals who are interested in seeing more of the data in bulk. There's a feature here to check your account information, as I said, to change your password or to be sure of what access credentials you have. Right now, for me and for the duration of the tutorial, this will be the professional edition. I don't know if you can see that I've got my GoToWebinar content here. There's a notification window, which is another way that we communicate with our user base. Right now it's showing that I have a notification. If I click on that bell, I can see the latest notification there. So that's what I wanted to talk about in terms of a landscape and getting everybody comfortable with the login credentialing process. And what I'd like to move on to for the last part of this webinar is the many use cases that I want to highlight to give everybody a sense of how best to apply the Mastermind search engine through the web interface for their particular clinical or research questions. So I'm gonna walk through these cases individually. There might be a little bit of overlap, but I tried to orchestrate the cases so that anybody who has a particular interest in one aspect of the Mastermind database should undoubtedly recognize which of those I'm describing when I call them out. And it's also staged so that we start off with pretty lightweight use cases or searches. And as you'll see, we get into the more complicated searches, and I'll highlight which one is the most frequently used by our user base, which we'll spend the majority of the rest of the tutorial talking about. So again, I have about 10 use cases. I'll go through some of them very quickly and others in more depth, but that should take about a half an hour here and before we wrap up and adjourn. So the first use case is, imagine if you are a researcher or clinician interested in a disease and want to know all of the associated genes. Some of you may not have to imagine that because in fact that's why you're here, but I'll begin by executing a search from this Mastermind homepage by typing in a specific disease of interest. So as I free text type in the disease window here, what you'll notice is that there's a series of drop-down selections The blue indicates those medical subject heading or mesh terms that I described earlier. And the gray is a reflection of the synonym or the reason why those were prompted in your drop-down window based on the mesh synonym library. In this case, I was trying to type in myeloid leukemia, and what I need to do now to begin my search is engage that term of interest from the drop-down window, and now it becomes live, and I'm able to search on that by executing the search capability here. And so what you can see on the left is that disease. On the right are all of the various genes that have been recurrently associated with that disease by Mastermind's indexing of the medical literature. And in the middle is the number of articles that talk about those two terms, the disease, in this case myeloid leukemia, and any one of those genes on individual rows in any one of these papers. So it's organized here in descending order of the number of papers that co-mention those two terms or any one of their synonyms. And what you're seeing here are, for those of you familiar with hematopoietic malignancy, a handful of very well characterized actors in hematopathology, such as MPM1, FLT3, and c-KIT, and one of the most highly ranked is ABL, the ABL gene. Also on this list are genes such as CD34, which I'm highlighting here, and I'm calling that out to indicate that this page does not necessarily indicate that the gene and the disease are associated by virtue of mutations in that gene. If you're not familiar, CD34 is a well-known biomarker for hematopoietic stem cells that are a useful treatment in the context of hematopoietic disease. And so CD34 appears recurrently in the context of myeloid leukemia in the sense that it's a useful aspect of the treatment strategy for that disease. So to determine whether there's a pathogenic mutation-related association between the gene and the disease requires a deeper dive, which I'll talk to in later use cases here. But you can see how Mastermind exhaustively has indexed the medical literature, and in this prioritized way will showcase all of the genes that have ever been associated with this disease of interest. So just to orient you again to some of the functionality of Mastermind, if you'd like to go back to the home page, you could click on this upper left icon for Mastermind. If instead you'd like to change your search in this window itself, you can delete your previous search and begin typing again, and then just hit the search window here. But I'll go back and begin on the landing page just for continuity here. So let me refresh that page and start talking about the second use case, which is to say, I am a researcher or clinician interested in a disease and want to know all of the associated variants in the context of that disease. So I'll use a different disease here. I'll begin by typing in, just as I had before. My disease of interest in this circumstance is hereditary hemorrhagic telangiectasia. I see that appear in the drop-down menu, and I engage and perform my search. And similar to what we saw before, I see all of the various genes that are recurrently associated with this disease in this number of articles. And not surprisingly, at the top, for those of you familiar with this disease, are genes well characterized to be associated pathogenically with that disease. Genes like ACVRL1, endoglin, inhibin E, SMAD4, ALK, etc. So if my question originally was, I want to identify all the variants that are recurrently associated with this disease, to get at that information, I would click on any one of those row elements, and then there's a lot of content here, so I want to focus your attention on the left panes. The top pane is a visual representation of the variant landscape of this gene in the context of that specific disease. And then all of that information is listed below by gene, by variant at the protein level, and the cDNA positions associated with that variant, and the number of articles that talk about that variant either with filters or without filters, which I'll talk to in a moment. And so to get at the question of what are the variants that are recurrently associated with that disease, you'd begin at the gene level and understand which genes are recurrently associated with the disease, and then iteratively go through each of those genes and collect all of the variants, as you can see here, by investigating the literature, which I'll walk through here in a moment, which is showcased in the user interface on the right. So this is something that our users do routinely if they have a handful of genes at most to comb through and look for variants and ascribe pathogenicity or treatment approaches for each of those variants. What we found is that there are users who have more high throughput needs and have a bigger palette of genes or more diseases that they need to investigate, for which Genomenon offers service projects that can be crafted with a specific goal in mind after a conversation with our clients. So let me go back to the home page. I'll clean out the previous search and introduce the third use case, which is to say, I am a researcher or clinician interested in a gene and want to know all of the associated diseases with that gene. So this is basically the converse of the search that I had performed initially, where I'll begin by typing in a specific gene, just in the same way as I had done with the disease. And you can see I've typed in EGFR, and there are various prompts, but the gene I had in mind was this first one here. So I'll click that, and then I can perform the search. And now you'll see on the right side, the gene is EGFR. That's my focus here. But on the left, I have a whole menu of different diseases that are recurrently associated with the EGFR gene. And the number of times that those two terms, the disease and the gene of interest, recur in the entirety of the medical literature. One thing to point your attention to is this all diseases entity. In the context of professional edition users, all diseases is the maximally sensitive search that can be performed. That is to say, the user clicking on that row would be taken to the Mastermind content for EGFR, irrespective of the mention of any particular disease or really any disease at all. That is a way to maximize the sensitivity of your variant level curation activities or research. Below that, there is a step off typically of about 50%, where the most frequently cited disease in the context of that gene is mentioned, but so too are all of the very many other diseases that are mentioned in the context of that particular gene. So if you want to enhance the specificity of your search, you can click on any one of these specific diseases and see the variant landscape in the context of this particular gene. But if you just wanted to get the information for the diseases that are recurrently associated with your gene of interest, this is the whole menu of diseases in descending order of their frequency of recurrence in the medical literature. Let me go back to the home page and type in a different gene. This is a P16. For those of you unfamiliar with that gene, its synonym is CDKN2A. That is the more conventional or standard gene symbol for that gene. If I click on and engage, then what I'm taken to is the same palette of diseases, obviously just specific now to CDKN2A. So let me go back to the home page, clear out my search, and then start in on use case number 4, which is similar to use case number 2, just going at it a different way, which is to say I am a researcher or clinician and I'm interested in a variant and want to know all of the associated diseases from the medical literature with that variant. So I'll begin just as I had before for P16 and EGFR, typing in the gene of interest. In this case, it is the c-MPL gene, which is a surface receptor in hematopoietic cells. And I have a variant that I'm interested in understanding whether it's been published before, and if so, in the context of what particular diseases. So you'll note that I typed in P dot and then the variant itself, and what dropped down is this suggested Mastermind key for that variant, R43X. I want to emphasize here when you're typing in your search for a variant that you can use any nomenclature that's convenient for you, whether it be conventional HGVS protein level nomenclature, such as I've typed in here, or otherwise using the 3-letter amino acid code or cDNA nomenclature or RSIDs, for instance. What will always need to happen is you engage this from the drop-down menu so that you can now execute the search into the Mastermind database, organized as it is around this variant key. So my search now is on the MIPL gene and this particular nonsense variant in the MIPL gene, and I'm going to perform the search. And what you see should be familiar by now. The variant is the same on the right in every one of these rows. The topmost entry is the most sensitive search. There are 7 papers out of the 30 million in PubMed, including those that were published just this past week. Out of those 30 million, there are 7 references that talk about this particular variant, of which 5 talk about the disease congenital amegakaryocytic thrombocytopenia, which is the most frequently co-cited disease in the context of this variant in 5 out of 7 of the papers total. And then some fraction of those 5, there are other diseases such as aplastic anemia or frank leukemia that are also discussed in the context of that variant. And so again, depending on your focus, depending on the number of search results that appear and how much energy and time you have to devote to your search, you can either cast a very wide net and see all of the papers using the all diseases, or otherwise go specifically into addressing your particular disease gene variant association of interest. Let me go back to the home page and highlight a couple of other different types of variants that can be performed in our search window. So this is a frame shift mutation. The lysine at 468 is involved at the DNA level in some frame shift, deletion, or insertion. And this is the Mastermind key that you would see if you typed in the C dot change or otherwise any permutation of the conventional HGVS nomenclature for that frame shift mutation. But again, you'll have to engage from this drop-down menu the particular variant that you've agreed with here in the Mastermind prompt before you perform your search. Similarly, I've chosen ATM because it's involved with a number of non-coding changes. If you have a non-coding change in mind, what you'll see is a drop-down prompt here where you've got, I think it's asparagine at the 2543 position, which is a reflection of where the cDNA change is in a splice acceptor, which is what the SA stands for. So again, we're showing you what the Mastermind key is for that result, and then reconciling that with your original search. So this is the way that you are able to search at the variant level using, again, any nomenclature that's convenient for you. After you've recognized the Mastermind key that's most appropriate, you click on it, then engage your search. So let me clear out that search and begin on the 5th and most frequent the use case that I'll spend the most time discussing to highlight some of the features on that terminal page that we looked at in the previous use case. So use case 5 is, I am a researcher or clinician interested in a variant and want to know if it's been published before and to understand about that publication landscape. This is by far the most frequent use case for our Mastermind users. There are many others that I described earlier and some more that I'll describe that are a little bit different than that, but this is the most frequent use case where previous to coming across Mastermind, users would either be searching through PubMed or Google Scholar. Mastermind is an excellent replacement of those processes in your clinical or research workflows that gives you vastly superior results in terms of sensitivity and specificity. And so the specific example that I want to walk through is a variant in the UROS gene. It is a missense variant. I'll type in this variant. As I said, if this is the variant that you've encountered in your patient's VCF file or otherwise in your research cohort, or if it's a variant that you have questions about or interested in for other reasons, typing in the gene and then the variant and performing your search will take you to this result page where you can get a landscape view of the diseases that it's associated with. And if you weren't familiar before, it should be apparent now that the UROS gene and this particular variant is associated frequently with porphyrias. We don't need to go into the nature of that disease, but you can see a number of aspects of porphyria and the different types of porphyria that are involved in this particular circumstance. If you want to maximize the sensitivity, you execute the all diseases search. If you want to enhance or hone the specificity of your search, you'd click on any one of these specific disease entities below. In this example use case, it's most typically the case that our users will engage the all diseases search, even as it does contain about 70 total references where this particular variant is described in the medical literature. I'll show you ways that you can hone the specificity of your search so that you don't feel the need to look through all 70 of those references to come to conclusions that you need to for your clinical workflow. So we talked about on the left the variant landscape for a gene-specific search. In this case, this is now showcasing those variants in the diagram that co-occur in the context of the mention of the particular variant that you've searched on. And so what you're seeing is all of the variants that also appear in the context of this particular variant and those references that mention the C73R mutation. Below is the comprehensive list of all of the variants in the UROS gene, just so that you never lose sight of the comprehensive landscape of all of the variants that Mastermind has identified. And we'll talk through a couple of those features here in a moment. But to turn your attention now to the new feature that we haven't talked about yet, on the right side of the Mastermind interface, where on the left side is the variant landscape, on the right side is the publication landscape. And we've depicted it visually here and in list form, just as we have done for the variant landscape. In the upper right is the literature landscape by publication date on the x-axis, and by impact score for the journal on the y-axis. Each of these individual icons represents a specific reference. When I hover over, you can see the title of that reference and in parentheses the journal. The size of the icon is a reflection of the specificity of that content to your particular search. In this case, we've not yet engaged any category terms. We've searched on just the gene and the variant. As you can see in these filter icons, we're casting a wide net for all diseases. We've searched on a gene and a particular variant. As I'll show you in a moment, when we add new category filters, you'll see these icon sizes and therefore the relevance of the content to your search change in accordance with what you've told Mastermind you're interested in seeing. What you can see below is all of that content, all of those 70 some articles that talk about the variant of interest that are visually depicted here. You can zoom in and reset that graph as you please. These are all listed below in the article list pane. If you're interested in collapsing the publication history that's depicted visually, you can collapse that and see a bigger sweep of the articles in list form below. These PDF icons indicate that Mastermind has secured and indexed the full text content for each of those references. So without going into any detail, you can see that Mastermind has a very broad reach into the medical literature. The journal for each of these individual references is depicted, as is the publication date. The title is depicted here, and so too are the number of mentions of the gene of interest and the variant of interest, which is a useful way for you to triage the relevance of the paper depending on your specific type of inquiry. At the top here is the ability to export this PMID list as a comma-separated variant file. That will download for you all of the bibliographic information and preserve the sort order of these references for those of you who want to get the information and do some downstream processing with it and don't want to transcribe this manually. There's the export capability. We have 2 link outs in partnership with Safator, who is producing the Varsum database. When you have a variant of interest and you'd like to see some population frequency information or computational models of pathogenicity, we have link outs to go to that information in the Varsum website through our partnership with Safator. And similarly, if you'd like to perform a search through Google, there is a Google Scholar link out that would include the various ways that that variant can be described so that you can perform a search in Google Scholar. But Mastermind has the content that's required to answer the questions that you need and is a much more thoughtfully organized and clinically relevant presentation of the information for your variant curation activities. So when I click on any one of these references, either in the list form or in the graphical form here, if I click on any one of those, you'll see 2 drawers open up from the bottom. On the left is the title and abstract of the paper that you've selected, and on the right are full text sentence matches for that particular paper that are highlighting the mention of particularly relevant terms from that paper. In this case, it's defaulting to genes and variants, but if you want to see only those sentences that mention the particular gene, you can click on that window. Or in the case that I want to highlight for this use case, if you want to specifically focus on the papers and their mention of the variant of interest, you can click on and engage the variants only window here, and then rapidly toggle from article to article to see all of the different ways that the author has described the variant, and more importantly, see the sentence context where that particular variant is discussed. So if I toggle through those papers, or if I identify a reference that I'm really keen to identify and interrogate, I can click on it directly or toggle to it. If, say, this particular sentence or set of sentences is particularly interesting to me and my clinical question, and I want to take a deeper dive, and I've installed a Google Chrome extension and have access to that content, you can see how this was presented through the web browser in the Mastermind interface just now. If you have access to that content, you can click on and see and interact with that PDF using a variety of these PDF tools. And so to get back to the sentence matches, you can click on Show Matches. To restore the panels to their original height, you can click on those Expand Pane capabilities. And that's the use case for identifying the variants described in any of these papers of interest to begin your curation activities. So a version of that use case is to say, how do I identify the most seminal paper, the original paper that talks about the variant of interest, versus identifying some of the more recent papers? Well, that's where this aspect of the publication history becomes meaningful. You can see well-supported, widely cited papers that talk about your variant of interest based on how high they appear on the y-axis, or most originally published or seminal studies on the hugging here on the x-axis at the left, or more recent studies, including those that were maybe published within the last week on the right side. Similarly, if you'd like to see that in content in list form, you can click on and engage a different sort parameter, not the clinical relevance or the content relevance order, which was what is presented by default, but rather the publication date order here, either in ascending or descending order. Another way to prioritize your search, if say 70 articles is too many for you to search through, and you're curating this variant in the context of the ACMG guidelines, a version of the use case that we're discussing now is, I'm a researcher or clinician interested in a variant that I've identified in a patient, and I wish to classify that variant or interpret that variant according to ACMG guidelines. So the way to do that for professional users, uses this third aspect of the Mastermind interface, which is the categories, a way to enhance the specificity of your search at the keyword or category level. And because the use case that I'm talking about now involves specifically curating for ACMG interpretation, I will click on that category header for ACMG, and you'll see a full palette of different category headings. In this case, these are specific to or focused on the ACMG guidelines. For those of you who are familiar with those guidelines, some of the most meaningful content that needs to be extracted from the medical literature involves designation of strong evidence for pathogenicity in the context of functional studies. So I click on functional studies here, and you can see all of these different key terms that are associated with functional studies, broken down according to whether they're in vivo or in vitro type assays or terms associated with functional studies. And I can either click on these individually, such as I'm doing here, click on them categorically, as I've done here, or enable every one that you see on the page entirely. And if I've enabled every one of them or a handful of them, and you are able to cross categories here as well, so this becomes very combinatorial, I submit with my chosen filters, and what happens is the papers from that original list of 70 that do not talk about any of the terms that I've engaged will be filtered out, but the papers that remain will be reprioritized, as you can see here in the publication history, as well as notice here in the context of the list, they've been now reprioritized with functional studies and the associated keywords as a primary focal point. And so to identify which of these references is the most meaningful, you can click on each of those references in succession and get a sense for the content, either the sentences that describe the variant, the sentences that describe specific key terms that you've engaged, in this case, functional studies, so things like wild type and in vitro will appear, or otherwise looking at all of them in their entirety, you can see the keywords, the variant mentions adjacent to each other and get a sense for what the authors were attempting to describe in this paper. And then again, if that piques your interest and it addresses a focused clinical question that you're interested in, you can seek out the PDF if you've installed the Google Chrome extension. If you would like to rapidly toggle through this information, one of the more meaningful ways to do that, initially at least, is to focus on the variant sentence contexts, is to focus on that in the drop-down at the right in the lower right pane, and then to very rapidly toggle through and read the information until you've identified which of those papers is most likely to contain the information that you need. In this case, this particular paper is meaningful because it has a number of mentions of the variant, and when I toggle to the keywords, it clearly is describing murine models of this particular porphyria disease that I'm interested in for my patient. And again, if that is something that you've identified as meaningful, you can click on and view the PDF if you have access to that content. Moving forward and out of the UROS gene to highlight some other features for a particular additional gene, if your question is, I'm a researcher or clinician and I can't find any literature on a particular variant of interest, how do I find information on a neighboring set of variants? So for that purpose, I'm going to the ATM gene. You can see the whole landscape of variants in the ATM gene in visual form here and in list form. If you're interested in identifying variants that are neighboring, if you want to tease apart some of these additional variants, say you have a variant in this region of the protein and you're interested in finding the variants that are neighboring, you can either do that by clicking on and engaging any one of these neighboring variants, or otherwise sorting, say, by cDNA position, as I'm doing here, and then focusing in on the region of interest, or otherwise filtering so that you're only identifying, say, loss of function variants, in this case, frameshift variants by typing in FS in the filter box, nonsense variants, as I've done here by typing in X, splice changes, splice acceptor, splice donor, or intronic changes, as I've done here. And once you've identified the variant of interest, you can click on the variant that's neighboring your variant or similar in its nature to the variant that you have in your patient, and then investigate the literature in much the same way that we had done with the previous example. So that was the biggest use case. I've got a couple more use cases that we'll go through much more quickly. They're less frequent, but still very meaningful, depending on the circumstance for the user. And so I'll walk through 4 more use cases very quickly. This particular use case is I'm concerned about copy number variants. So this use case involves being a researcher or clinician interested in a gene and wanting to know if it's associated with a copy number change. So I'll perform the gene-based search. I'll cast a wide net with all diseases, although focusing in on a particular disease works just as well. Since you're interested in copy number change and this is not properly a variant, you would go to the genetic mechanism pane, engage the copy number subcategory here, and what you can see is a palette of key terms pulled from the full text of, in this case, 153,000 references that talk about EGFR. In this case, for those of you familiar with EGFR, you might know that exons 19, 20, and 21 are particularly meaningful. If you want to see any of the sentences from any of the papers that talk about EGFR and any one of those exons, as well as in the context of, say, deletions, you would engage those key terms and perform your search by submitting with those chosen filters. You can see those filters appear at the top here in the context of deletion. If you say want to focus your search, that's perhaps too many, and your patient just has this exon 19 deletion. You've got now exon 19 and deletion as key terms. That's still quite a number of papers that talk about those terms, but they're prioritized here. If you click on any one of those references, and in this case, focus on the sentences that talk about any one of those particular key terms, you can very quickly get a read on what the authors are describing in that context. So that's one way that you can get to the copy number change information in Mastermind. Another aspect of the searches that our users have brought to us is, say, they're interested in fusion events. So this use case is I'm a researcher or clinician interested in a gene and want to know if it's been associated with a fusion event in the context, say, of a specific disease or otherwise in any context. If I just broadly say I'm interested in ALK fusions in the context of lymphoma, I perform that disease gene search as you see here. I'd go back to the genetic mechanism pane and click on the fusion events, and in this case I would enable all, and I would submit with my chosen filters. And one of the first things that you see here, if you weren't familiar with this disease in the keyword sentences, is this EML4 ALK fusion. And if I were to toggle very rapidly through all of the papers that talk about fusion events in the context of ALK and lymphoma, you'd see EML4 ALK recur. And if now I'm interested in specifically focusing in on those papers that talk about EML4 and ALK in the context of fusion events, I can switch to advanced search, as I've just done here, which allows you to perform multi-parametric searches. And in this case, if I'm interested in typing in, searching for EML4 in addition to ALK in these papers, there's a Boolean operator here that I can toggle. So OR would be inclusive of any mention of either of those genes, and would be the overlap between mentions of ALK and EML4. If I perform that search with the fusion event filter, now I've really focused in on and prioritized the references that are most relevant to fusion events for EML4 and ALK. And again, if you're interested in the most seminal paper that talks about that fusion event, you can see that the EML4 ALK Nature paper, in this case in the context of lung cancer. So similarly, if I refresh that page, go back to my original homepage, refresh, I can switch to advanced search, and for one of the penultimate use case, if I am a researcher or clinician interested in 2 variants from a patient and want to look for compound heterozygosity ever published in the medical literature, I can perform a specific variant search in the advanced mode by first engaging a gene and next engaging a particular variant for that gene. In this case, the example that I'm using is trivial. It's KRAS G12D and KRAS G13D. Now I've got the AND operator in my advanced search capability. KRAS as a gene is necessary to appear in the paper to ensure that it's in fact a KRAS variant. If I'd like to perform the search, what I'm now seeing are both of those variants are present in 1,300 individual articles in the all diseases category. If that's what I want to focus my search on, because I want to cast a very wide net. If I'm particularly interested in, say, compound heterozygosity in the context of a constitutional disease, I can go to the ACMG classification category and identify where compound heterozygosity appears as a key term and perform that search. So again, that's in the context of a constitutional disorder. That would be a way for you to focus your search among those 1,300 articles that talk about both of your individual variants that you've identified in your patient. And that's not necessarily just possible with the same gene, but it could span multiple genes. So I could have just as easily performed the search for a KRAS and an EGFR variant in advanced mode, as I've just shown. So the last example that I want to walk through, again, is an example that has come to us from different users. The use case here is I'm a researcher or clinician interested in a variant from a cancer patient and want to determine what drugs have been used to treat that patient in the past. So in this case, let's just keep it very high level. If my variant of interest is V600E and BRAF, let's say for example's sake, I'm interested only in melanoma patients who've been treated with particular compounds targeting this BRAF mutation. The place in the categories to get this type of information is one of 2 places. The first is if you don't know what you're looking for, the significant terms category here will highlight a number of different terms that are recurrently mentioned in the 4,500 articles that talk about BRAF V600E in the context of melanoma, many of which you can see here are specific compounds. So if I highlight those after picking them out from this palette of different recurrent key terms and perform my search, that will filter those 4,500 articles and prioritize them in list form to focus in on the content that most specifically talks about those different treatments. Similarly, if you already know what term you're looking for and you'd like to perform the search explicitly, I'm switching now to advanced search. If I say I'm interested in vemurafenib, I can perform the search by typing in free text in the abstract search here, and now my focus from those 1,500 articles is specifically on articles that talk about vemurafenib in the context of BRAF V600E mutated melanoma. So I understand that we're running up on the end of the hour here. Just to highlight and top line the most common use case for Mastermind would be to say I have a variant of interest. I'll go back to one of my previous examples. I perform the search for that variant. I cast a wide net on all diseases. I engage from this window here. I choose what focus I want to bring to bear for these papers. In this case, say it's the variant sentence mentions, and I can quickly read the titles and abstracts of each of these 7 papers as I rapidly toggle through and identify which references are most likely to address my clinical question. So this last example here is the most efficient way to get at the information that's most likely to be relevant for your variant interpretation needs. So that concludes the tutorial. I want to thank everybody for your presence and attention. to highlight again that you have free access, even if you're not a paying user of Mastermind, indefinitely. So free access throughout. For new signees, you have professional Mastermind for the first month, and then you'll revert to free. For those individuals on the webinar here who have more advanced needs, things like automation capabilities or large projects in mind, we have API capabilities that we can discuss, as well as performing service work. And what I'll leave you here with, if we don't have any time for questions, is please don't be shy. Please feel free to reach out to us either through the Contact Us form, which is ever present in your Mastermind interface, or by emailing support@genomenon.com directly. We really encourage a lot of feedback and interaction with our user base and our development and support team. So with that, I'll turn it over to Candace. And again, thank you all for your attention.
Thank you, Mark. Thank you so much for joining us today. I will be sending a follow-up email with the link to the recording in the next week, and we'll have the questions that were asked answered on that video page for you, but we don't want to go over today. So be sure to visit us at genomenon.com, follow us on all social media at Genomenon, and like Mark says, we love your feedback, and we love that you're a part of the Mastermind community. Have a great day.